Hello, I'm Jamie. I'm Director of Collections and Programming here at Thackray Medical Museum, and I'm here today with the fabulous bioartist, Anna Dumitriu.
Thank you, Jamie. It's a privilege to be here at the Thackray, and I just wanted to do a bit of a tour of the show. So we're going to go around and have a look at some of the objects and explain a bit more detail about them.
So let's start with this first piece, the star of the show, the plague dress. Anna, you're a bioartist. Tell us a little bit what a bioartist is and how that relates to this piece.
Bioartist work, it's a big subject because there's lots of different approaches to it, but bioartists tend to work with biology and they work with biomaterials, so sometimes living matter. In my case, because I work with dangerous pathogens sometimes, I tend to kill those things before I exhibit them, so the traces of them are there. But other purists might have all living matter in it, but then they won't work with the dangerous pathogens in the same way.
So we can't catch plague from this?
We can't catch plague from it, but it has got plague DNA on it, so the extracted DNA of Yersinia pestis, which I extracted myself in the lab at the UK Health Security Agency in Colindale, which is the laboratory of the National Collection of Type Cultures, where I have a residency, an artist's residency, in the working with them. It's the oldest collection of pathogenic bacteria in the world.
One of the really fabulous things about being here in front of this dress is it's not only wonderful to look at, but the smell is amazing. Tell me a little bit about the
it's stuffed full of lavender and spices, and these are the things that they would carry to ward off plague back in the Great Plague of London time. So the whole thing's inspired by that. It's a silk dress because it's raw silk, because the plague actually travelled from the steppe region through the Silk Road, through the maritime Silk Route, down from Canton or Guangzhou, as it's now known, down to Amsterdam and then on to Britain. And so it's about that. And actually the first place that this piece was ever shown was in Guangzhou. And then it's also been shown in the Netherlands. So it's kind of tracing that route, so it's now on show in England. And it's dyed with walnut, which Culpeper, the herbalist, said he thought was a treatment for plague. And it's a 1665 dress design, but the embroidery on it is actual period embroidery from that time. And that's where I've impregnated the DNA.
So tell us a little bit about this piece, Anna.
So this piece is called Zenexton. It relates quite strongly to the plague dress. It's an amulet. So I was very inspired by the idea of Paracelsus, who was a sort of medieval physician, talked about how that we used amulets to ward off diseases, but the amulets were kind of, if you think of them a bit like antibiotics now, they were kind of broad spectrum antibiotics, so they would be warding off disease in general. But he said that plague was such a big problem that we should just we should have a specific amulet that would ward off plague. And then I was fascinated by the way that the debates then go, because he said, I won't come up with this in my lifetime. It's for the natural philosophers or the physicians of the next 100 years or so to come up with. And so then this debate starts about what it should be. Now they sort of kind of agree that it should maybe have a toad in it, because they all kind of agreed that toad was very good for warding off the plague. So they started off with the idea of hanging a toad down your neck. So this shape of it sort of looks like a heart, but it's actually a kind of the back of the toad with the head of the toad shape. Is it with these sort of warts and things of the toad skin, referencing that. And then over time they're sort of like, well, it should be paste of toad. No, it should be powdered toad. And it reminded me of how we talk about sort of using gold in things or nanotechnology and stuff like that. So it's kind of, if we made it smaller, it would be more effective. And then he says it should probably contain sapphires, because everyone knows that if you put a sapphire onto your plague bubo, it will turn black and it will leach the disease from the body. So it's got sapphires on it. These are lab-grown sapphires. Then the debate goes, well, it should probably contain menstrual blood, because they thought that was very good for amulets. And then well, the menstrual blood of a virgin. No, the first menstrual blood of a virgin gathered on a full moon. So we've got the drips of blood, and we've got the moon shape there as well. And it's hung on a ribbon which is dyed with walnut, the same as the plague dress. But this, and he called it the Zenexton, and I haven't been able to find any reason as to why he came up with that name or what exactly it means, but I love the word. It just really fascinated me. But the idea of this piece is that it kind of achieves Paracelsus's dream, is because I worked with Christine Rollier, who's the professor of vaccinology at the University of Surrey, who was previously with the Oxford Vaccine Group at the University of Oxford. And she has now a plague vaccine based on the Chadox 1 platform, which people might remember was the famous Oxford AstraZeneca COVID-19 vaccine, was also built on this platform. She's built a plague vaccine on this platform, and it's inside the necklace. So it really does ward off plague.
So how important is, it seems like it's a theme through some of your work, certainly in terms of what we were just talking about with the plague dress, the idea of linking the past and the future.
Yeah, absolutely. These sort of threads across time is really important to me, and I think we can learn a lot from the past. So we kind of repeat a lot of things, and then some things we did then that sound crazy, like toads or whatever, actually might not be as crazy as we think, or might have kind of techniques that we still do now. And then I've done quite a lot of work about alchemy as well, because I'm really fascinated in how the rhetoric of the alchemists actually really fits with kind of synthetic biology and stem cell therapies and things now. So it's actually, I think we as humans are still very similar to what we were, and I'm really interested in looking at how that will play out in the future. And I should mention that the necklace is 3D printed metal. I worked with Alex May to design the casing, and then it's been 3D printed in brass. It's gold plated. So it's quite a modern technique to achieve it in the end as well.
So this is a really interesting piece, and I think for me speaks a lot about some of your work in terms of that we think often of germs and bacteria as quite dangerous, quite unknown kind of things that can do us harm. But I think in this work, this very much speaks about the care and protection that germs and bacteria give us. Can you tell us a little bit more about that?
So this piece is called Bacterial Baptism, and it's the idea about the moment when you're born and you gulp down loads and loads of your mother's microbiome, the bacteria that live on your mother, which seeds your gut microbiota, your skin flora. And so it's called Bacterial Baptism, that's why it's a christening gown. But it was inspired by work that Jane Freeman was doing, one of my collaborators, and she was studying how Clostridium difficile bacteria, now known as Clostridioides difficile bacteria, which is considered to be a dangerous superbug that affects people when they take a lot of antibiotics, certain antibiotics, and it can kill off the gut microbiome. So that you get this situation called dysbiosis, and this bacteria, which may or may not live in people, it can colonise all those gaps that are left by the killed off bacteria, and it makes people really ill. But it lives in children. They first discovered it in babies, newborn babies, and it doesn't, as far as we know, it doesn't cause them any problems. It's called C. diff. Because it was difficult to grow, not difficult to cure. And it wasn't until the 70s that it actually became a pathogen, really. It was just part of our normal flora, because it picked up antibiotic resistance genes, which meant it could resist the drugs that we used to treat it, the drugs that were killing off other bacteria in the gut. It could basically fill up those ecological niches. So it might be that people caring for their grandchildren or something like that might pick up this bug, doesn't cause them any problem, and then when they're exposed to broad spectrum antibiotics, it might cause a problem because it can recolonize them. So it was about this kind of moment, and the original paper, which we referenced by Hall and O'Toole, which was a paper from the early 20th century, talks about how they were trying to prove that it was a pathogen and things like that, and they were growing them in rabbits and guinea pigs and things. So I've got these like guinea pigs and rabbits embroidered on there, and I've got the bacteria embroidered on there. The other embroidery that's coloured embroidery on it is with some of the dyes that they use to dye bacteria, and these kind of pendulum bits here were actually embedded in what they call a gut model, one of these bubbling bacterial ecosystems that they use to study how the gut bacteria works outside of the body, which we've got more on that later in the exhibition.
So in terms of like antimicrobial resistance, it's a live kind of issue that affects people. How much and how important is it for your work to reflect some of those?
Yeah, really. That's why the title of the show is Fragile Microbiome, because I'm thinking about, so the microbiome is kind of the name for all the bacteria and all the other species that live in us and on us and around us. It depends which microbiome you're looking at, really. So you could look at a whole ecosystem as being a kind of microbiome, but in some cases we're looking at the gut, and some cases we're looking at humanity as a kind of ecosystem. And antibiotic resistance means that when bacteria pick up these genes, and they can share these genes that allow them to resist certain antibiotics, it means that they're not treatable with those drugs anymore. So people that get those infections can't be treated. And in people that are on like chemotherapy or have operations or something like that, it could be devastating potentially. So the scientists I work with are working very hard to try and find out and understand about this problem, like how are these genes spread and shared at different levels in the bacteria, and what can we do to combat it. And a lot of it isn't so much to do with finding new antibiotics as finding like the genetics of it, understanding the genetics of it and how those genes are spread, so you can track it better. And if you can track it, then you can stop it spreading, in theory, at least. And I mean, in talking about the plague, which we know killed, a third of the population of Europe, at one time. They've actually found antibiotic resistant plague in Madagascar, where there's been an outbreak. I'm not saying that we're going to have this massive influx of that and that's going to happen again, because it's not a massive resistance. It has resistance genes rather than being actually resistant, but it has those genes. Which is a risk, obviously. So even in terms of these dangerous pathogens, that could be a big thing. But also in terms of, a kind of bacteria that can cause a pimple, that could actually kill you if it gets into your system. Or dentistry, like people used to die in the plague era. When you look at the bills of mortality, a huge percentage of people are simply listed as dying of teeth, and that was infections from their teeth. So it was still bacterial infections. This piece is called Spindle, and it references how Clostridium means spindle, and they're spindle-shaped organisms. So these things, these sort of pendulums, I think of as spindles. It's crocheted linen lace, and they were grown inside the gut model. So I crocheted them, and these bits that are very brown, they're quite hard, quite solid. So they've actually had to be, because it forms biofilms, and biofilms allow it to kind of swarm over the gut, like layers and layers of bacterial ecosystems. And it also forms spores, so it makes it very hard to treat because the antibiotics that do treat it can't get into the spores very easily. So you get these layers of the biofilms and the spores. So this took a lot of killing to be able to exhibit it safely. So it's been autoclaved, which is a high pressure steam sterilisation on a destroy cycle, they call it, to make it safe to exhibit. But that's why it's referencing this idea of the spindle, and also Clostridia. We'll go over to this one here. This idea of the spindle, it comes a lot from Sergei Vinogradsky, who was a pioneer of microbiology. He worked with Pasteur in around the 1880s, but he was kind of considered a madman at the time to an extent, because he thought that you couldn't look at bacteria in isolation. So Robert Koch and Louis Pasteur were looking at, like, does this germ cause this disease? You know, and they were doing things like taking it from a pimple and putting it into a guinea pig and seeing what happened to the guinea pig, like quite disturbing things like that. Whereas Vinogradsky was looking at whole ecosystems, and he wrote a book called The Microbiology of Soil, and he, like, this interconnected, heavily interconnected ecosystem, and he became obsessed with the retting of flax. Flax is the plant that you use to make linen clothes, which is the weirdest process in the world, because they get the plant stems, bind them together in a bunch, and put them in stagnant pools of water where the bacteria act on the plant stems and make it kind of peel apart, and then they get rid of the hard bits and just keep the soft bits, which are then combed. And this is made of that. So this is the raw flax, and it's kind of woven together. And then we've got the bacteria, one of the bacteria that he discovered when he was doing this research, which is called Clostridium pasteurianum, it's called. But they changed the name to pasteurinum. We don't exactly know when, and we don't know whether it was Vinogradsky who termed it spindle, you know, Clostridia, or whether it was just, people just say, oh, it's because it's spindle shaped, but he was working with linen. So it seems really interesting that it could be interconnected. And so this Clostridium pasteurianum means spindle from the field. So that, and this is one of the ones he discovered when he was looking at the flax plants that were rotting. And they still do it. So if you're wearing linen, anybody listening to this is wearing linen, it wasn't maybe in a stagnant pool, but they use these big tanks, and they have bacteria that they put in the tank, and it's the same process. So even now, to make linen, it's made with bacteria. So you're really looking at a biofabric, but an ancient one, because as far as we know, the ancient Egyptians first discovered linen for clothing, and then they would dye it with things like turmeric. So it was considered to be antimicrobial, and holy people or whatever would wear this, and healers would wear these turmeric clothes and turmeric dyed linen, which turmeric is antimicrobial, and obviously linen's made with bacteria. It's much more kind of tied up with our world than we think about the bacterial world. So inside the beads, the purple beads, is the Clostridium pasteurianum, as it's now known, which I got from a German bacterial collection. There's some Clostridium difficile, Clostridioides difficile bacteria there, some flax seeds, and referencing that idea of the flaxen haired maiden, which is a bit like that, is actual hair. These flowers on here are hair wreaths, which is a kind of what they call a mourning craft. When people in mourning, they would make these flowers from people's hair. So this is incorporated in it as well.
So this case, someone with a little bit of a fear of going to the dentist makes me feel a little bit uncomfortable. Yeah, I can really see the beauty in it. They're such kind of beautiful works. How do you sort of balance that dichotomy in your work of people feeling uncomfortable but also really appreciating how beautiful these objects are?
I mean, I'd sort of say that that's the art happening. Like when you look at this work, if you've had these kind of slightly uncomfortable feelings and a kind of beauty that draws you in as well, that's kind of how I want you to feel, I think. Because people sometimes think that art should be something pretty or that it should be something dramatic and stuff. But we can have complex emotions about things that maybe we don't resolve straight away when we look at something. What we like is I've been talking to my friends and talking to my partner Alex about it, and what we talk about is this idea of like something staying with you and nagging you as well. So it's like, if you might remember it, and that's something good for me. So we've got these 3 works here, which are called, 2 of them are from a series called Teeth Marks, and it's that idea that dentistry has left marks on us. Especially historically. I mean, I talked about people dying of teeth in another part of this, and I focused a lot on that. This central work here is called Microbiome, and it looks like teeth, pulled teeth, but they're not actually, they're made of porcelain. But the glazes on them is a collaboration with Melissa Grant and Rachel Sammons. The glazes on the teeth are derived from the bacteria that cause tooth decay. So there's a brown, rusty coloured dye on it. And the central tooth at the front there is made from a biomineralizing bacteria that has the ability, when put in the presence of the right minerals, it will express tooth enamel. So we grew it over a kind of sponge clipped in the shape of a tooth, and then we sintered it in a kiln to kill the bacteria and just leave the tooth enamel. It's a little fragile, but the scientist that I was working with on it, she was almost told, like, people aren't going to want teeth made out of bacteria. That's like the opposite of what people want, is they kind of make them feel uncomfortable. But when we've exhibited this and we've done workshops with it, people are fascinated in the idea, because people are kind of getting used to that idea that we can use bacteria in other ways now, that they're not all harmful, they're not all out to get us. Some are, but these aren't. Is actually, they could be very useful at making things like that. We can use them to produce stuff that we need. It's a bacterium called Serratia N14 that's producing the bacteria, but maybe we could have the teeth of the future made from bacteria if we lose a tooth.
Fascinating.
And this idea of the past and the future comes together in this piece. So all this is a collaboration with Birmingham Dental Hospital, who have a big collection of historic objects about dentistry, which, working with Alex May, we photogrammetry scanned them, which is, you can take pictures all around and every side of it, and you can build up a 3D object from that, which you can 3D print. Birmingham Dental Hospital is one of the oldest dental hospitals in the world, arguably the oldest, although there's one in America that has a claim as well. And they built it right next to the Birmingham Jewellery Quarter because they needed the skills of the jewellers to make the false teeth and things like that. So that's why jewellery and dentistry is really linked. Birmingham Jewellery Quarter's kind of most famous thing they make is a charm bracelet. So what I decided to do was to make a charm bracelet. So it's an actual Birmingham assay charm bracelet there, but all the charms on it are 3D printed in solid silver from the photogrammetry scans that we made in the dental hospital from their collection. So you've got things like a narwhal tusk, which is about this big, which we scanned.
We have one upstairs.
A jaw opener, which is this bizarre thing for people with lockjaw to open their jaw, some false teeth, some tooth pullers, and even a very flat Anglo-Saxon tooth that they had because they had very gritty diets. So it was like these objects. But it was also linking to the fact that now their research is using photogrammetry. So they'll actually scan the inside of people's mouths, and they're now trying to 3D print at least the prosthetic bit, not the teeth, but the bit that goes, the teeth bit into. I don't know the actual term, but that is being 3D printed. So they're actually in the NHS, in Birmingham Dental Hospital, researching how's this going to work in the future, these new technologies. So again, it's that thing what we were talking about, the past and the future, and where we're going and what we can learn from it. And, you know, dentistry has moved on a significant amount. And that kind of leads us to the most profound mystery, as it's called, which is a term that was described by W.H. Mortimer. He was talking about false teeth. He said they were the most profound mystery because people wouldn't say that they were wearing false teeth. It was all very hidden. And fans were popularly carried because people cover their mouths. So these women looking sort of coquettishly over their fan, and they were hiding whatever was going on in their mouth, and they would eat their food from behind it as well, or they would eat their food in their bedrooms or something away from the others because they had false teeth. And people in those days didn't have very good dentists. They had a lot of rotten teeth. So this fan is made of bone, as false teeth used to be made. They would make them carve it out of animal bone. And it's an antique fan, so it was broken, so I've wired it together as they would do with gold wire. And then the fan, all the silk on the fan is a bit like this. It's grown in a petri dish with the bacteria that cause tooth decay and the bacteria that cause gum disease. And out of all the artworks I've ever made, including the artworks made with gut bacteria, that is the smelliest. I was literally spraying it with air freshener in order to work with it and stitch it together. It doesn't smell anymore because it's faded over the time of being shown, but it was quite an interesting aesthetic experience from my point of view as well in the making. And a lot of that happens in my work because the work's half made in the lab and half made in the studio. So it can go from working with dangerous pathogens in the lab, double gloved, whatever, with gloves taped to lab coats and things as you would with working with plague. I've worked with live plague as well as with the killed plague that was used to make that dress. And then it could be me sitting at home stitching things, washing the TV or something so as simple as that, or laying them out in the studio, working with them like that. So it's a bit of a mixture in my process, with my kind of aesthetic approach as well, I think.
So I think looking around this exhibition and looking at your work, it really makes me appreciate modern healthcare advances that we are. And this piece in particular was an item that really made me appreciate where we are in terms of the treatments and healthcare that we're able to provide. Can you tell me a little bit about this?
So it's a pneumothorax machine, but I've altered it. Actually, you have one in the collection just like this. And when I made this and I came here first time and I found the same one, I was very excited going around the collection. I was like, oh, they've got one. But this one I've altered. So I've carved it with the texture of how a very severely damaged lung might appear if it's been affected by TB. And I know I've got it right because I've had scientists, I did a conference for Médecins Sans Frontières in Tashkent, and had scientists from all over the sort of Central Asia going TB in the lungs. So they recognised that it looked like that. And the engraving on the cylinder there is how TB can look under the microscope. They call it the characteristic scattered red ribbons. So you see this red ribbons. But this pneumothorax device was it was part of the collapse therapy. So they would take the needle, attach it to that, and they would introduce air into the chest cavity to collapse the affected lung in order to give it a rest. And this is before we had the antibiotics that we can now use to treat TB. But there are parts of the world that can't get any access to those antibiotics. And in this country now, my collaborators at the University of Oxford have done amazing research of actually trying to find out what antibiotics will treat which person's TB by looking at the genome of the tuberculosis and growing tuberculosis in the presence of multiple antibiotics, and they have a huge data set. So now they're able to infer from that data set which TB can be treated with which antibiotic. So, and in the UK, everyone who has TB gets whole genome sequence. But in other countries, so this was what became very explicit when I went to the conference in Tashkent, was in other countries such as Russia, and Afghanistan and places like that, it's very difficult to get access to the drugs. And there were a lot of TB activists from Russia there who are fighting to get medication for people because it's really expensive. They don't have a general health service, so it's a huge problem there. And people won't go to the clinic because there's so much stigma to it, so they will literally be shunned by their community if it becomes out that they've got it. So even being seen to go into a clinic, if one pops up in your town, can cause great stigma. So they've actually done trials in the sort of last couple of decades with pneumothorax again in places like India to see if it's effective against multi-drug resistant TB, because that's another thing with antibiotic resistance. If we don't have the drugs to treat it, what do we do? And this did show some success in treating the multi-drug resistant TB, a technique like this. So it's not so far away from something that could potentially be happening even nowadays in some places. Going back to the history of it, we've got these tiny lungs. So I found some advice from, I think it was about 1902, from the British Society for the Prevention of Consumption, which is the old name for TB, because it literally consumes you, people waste away. And it said, where there's dust, there's danger. Halls and passages must be kept free of dust. And so they used to believe that TB was caught from dust, like you'd sweep up the dust and you'd inhale the bacteria and you would catch TB. You can't actually catch it like that. Generally it's caught by somebody with active TB coughing on someone because it aerosolizes the bacteria at the right level to be inhaled. So the main risk factor really is sleeping in the same room with somebody else with TB, like in a small bedroom. That's why homeless people in shelters can be very affected by it, because it can spread quite quickly. So this is it's made from needle felt, so it's wool, but mixed with dust that are gathered, and then it's impregnated with the extracted DNA of TB. So it's sort of where there's dust, there's danger. And it's really interesting, this idea of the dust, because when you think about TB and what is changed about society, that's where it gets so important, because things like modernist design, the staircases with no gaps, the idea that light should reach every corner of the room, big windows and light, that all comes from ideas about prevention of tuberculosis, because it was such a massive problem. And it still is. 2 million people or so every year die of TB. So if you think, from the COVID pandemic, it was something like 7 million altogether. This is happening every year. And between a quarter and a third of the world's population carry TB. And of that, 10% will get sick, and without treatment, half would die. So it's still a really significant problem that we're not doing enough about. It's quite good in the UK. We probably have some of the best treatment in the world for this, because we have the scientists who are very, concerned with it and working on it, and great facilities for genomics. But in other parts of the world, it's still a massive, massive problem. And that brings us nicely onto this one, actually, which is called coming back, because people say, oh, when you talk about TB, they say, oh, I heard it's coming back. And they say, well, it's because of immigrants bringing it over. So it's coming back because of the immigrants bringing it over. But what they don't know is how true that is, that it's coming back. Because the strain circulating in places like Bangladesh and India is called the Euro-American strain. And it's the strain that was taken over by the colonialists in the sort of the Victorian era, in the colonial time. And it's that strain that took over there because they didn't have the immunity to it, and it became the main, the significant strain of TB that was circulating. So this door is called coming back, and it's the idea you can't shut out disease. It's a global problem. It's like the idea, we have good care here for it, but we need to think globally. We're not just separate. And the COVID-19 pandemic obviously showed that very well. And I started this before the COVID-19 pandemic. So the paintings on it are the TB bacillus in the cells, as you might see it under a microscope, and the carving is like the pneumothorax machine. It's these granulomas and cavities that form in the lung when we're trying to treat it. And these parts are impregnated with specifically the Euro-American strain of TB. The DNA of it is in there. And this doorknob, which was my starting point for making the piece, and I found this doorknob. It's an antique doorknob from a TB sanatorium in America. And it's got this cross of Lorraine, the 2-bar cross on it, which the American Lung Association used as, they called it the crusade against tuberculosis. And we know the idea of the crusaders now is very much a kind of colonial thing as well. It's like going to places and trying to change the people there and convince them to be a certain way. And so it's interesting, this idea of the crusade there, and this idea that you can't keep things out. We need to think globally about healthcare, but with a good attention to different approaches in different places. I did a collaboration with a charity called Target TB, where they actually went into the communities they were working with and asked people in East Timor and another place, Ivory Coast I think, where they thought they got TB and what they thought caused TB. And somebody there, I mean, they came up with lots of different things, like eating unripe mangoes might cause TB or something like that. And one person said that they thought it came from cats, like cats could pass on TB. And they thought it was because of the purring sound. It sounds like they've got TB because of the way they breathe or the way they're purring. And you know, we all looked at this and thought, oh, that's silly, aren't they? A few weeks later, it came out in the press that a child in the UK had caught bovine tuberculosis from a cat. That had caught it from cattle. So it's not as strange as it seems, and we need to think about these like other cultures and their own traditions and approaches as well as we do this. Otherwise you become colonial again, or you become a crusader, and that's not a good approach to global health. No.
It's interesting just in terms of you talk about your work and you know, that it does engage with real live kind of issues that we grapple with today. And you know, certainly in terms of health inequalities, I think this speaks really powerfully of that.
Yeah, no, it's a really important thing throughout the work, is all these different issues and thinking, because I work with a lot of scientists who are working with global health and lots of different medics. So yeah, that's a big part of it. And some of these bacteria have been with us since ancient times, and we could go and look at this piece over here.
So tell me about this piece, and particularly the square I can see here.
So, as I was talking about, tuberculosis has been with us from ancient times. It's one of the oldest infectious diseases that we've been able to trace. Hansen's disease, Mycobacterium leprae, which used to be known as leprosy, is the other one. You can find that in very early bones, Pliocene era mammoths and things like that. And I did an Institute of Advanced Studies fellowship at the University of Surrey, and as part of that I was able to meet a scientist called Mike Taylor, who had previously researched, he was emeritus professor, but he'd previously researched looking for what's called ancient DNA in these very ancient bones. So he'd found bovine tuberculosis, which is related to the Mycobacterium tuberculosis called Mycobacterium bovis. He'd found that in Iron Age Siberian human remains from a burial in the Siberian permafrost. And I said to him, well, I would love to learn how to do that. And as I told you, I get really stuck in, hands-on. And he was like, well, I think that should be possible. You'd have to, a few days to coming here and working just on this. But it should be possible. I will write to the person in the museum where I got the previous bone samples and see if there are more bone samples we could get. He managed to get some samples of these Iron Age Siberian remains, and he taught me over a 3-day period the protocol for extracting ancient DNA from bones. And I said to him, what would you like people to know about about this, and he just said, just tell them how very difficult it is. It's really hard. It's not just like sequencing bacteria. It degrades so much over, centuries with UV and in interaction with all other different things there. It's really hard to locate it. So he was like, don't get your hopes up, it might not work. After 3 days, we were able to find a signal that we'd actually found some bovine tuberculosis DNA in these remains. So my experiment with him was successful, we found it. Now this is a bovine femur. So these were human remains that we worked with from the Siberian permafrost. But for the sculpture, this is a cow femur, so a great big strong bone, huge and ridiculous looking bone really, which the University of Surrey Vet School and Anatomy Department prepared for me. So actually bones are kind of disgusting and weird and leaky things, but this has had all that removed, so it's just the kind of the bit you can work with sculpturally. Otherwise it would be a very different kind of living art here, and it would be very leaky and quite smelly as well. And what I've done with it, I carved it so it would have the kind of the cavities and things that bovine TB causes in bones. So if you look at bones that have been affected by bovine TB, they've got these holes in it. But when he cut out the samples that we were going to use, he said, well, I'm going to cut them as a square. And then scientists or researchers of the future will know a human did this, a scientist did this, not the bacteria. And I thought, well, that's lovely, because it's like this mark of the scientist on the bone and on the past moving to the future. So that's why I put these squares in it. I carved these squares to be like the squares that he cut from the bone samples that we used. And then there's some painting of the bovine TB bacillus on it as well. And the carvings are all impregnated with the amplified bovine TB DNA marker that we found in the actual bones. So it's actually on here and in those carvings.
So how important is it, because again that's coming through in your work, that there's actual DNA from some of these bacteria that you're discussing, how important is it that they're actually an integral part of your work?
I think it's really important. There's something I really like, a sort of idea of truth to materials, and I like to have the truth of the stuff in there. Otherwise, it's purely a representation. I want to combine those two things, of it's the real stuff. It's authentic, and you're really close to it. I wrote a paper with Dr Nicola Fawcett, which was called "Bacteria on Display: Can We, Should We?", which was talking about even if there's a tiny risk, is it safe to do it? And we'll come to the work that I made with her that was around this that we were referencing, and she said that it's a thing. It's like when you go. I think she used the example in the paper of like when you go to the Hard Rock Cafe and you see somebody's famous guitar there. You need to know it's their guitar rather than just any guitar that they found and put the label on. You want to know there's something in that makes it. It gets you more, it makes you feel something more, and that's what we're trying to do with this, and that's how it works with me. I think I need to feel that, and I hope other people appreciate feeling that as well.
So I think a lot of people will recognise the phrase "make, do, mend." How does that relate to BioArt and your work?
Yeah, so there's a whole rhetoric around the idea of patching and repairing genomes of bacteria or genome editing, and there's a technique called CRISPR DNA editing, which stands for clustered regularly interspaced short palindromic repeats, which are fragments of DNA that reappear throughout all different kinds of species of things, and bacteria and human DNA. And people talk about, can you cut this out and can you patch it? And you need, sometimes depending on what kind of bacteria or kind of what organism, you need a repair fragment, because, for instance, human DNA will knit back together, but bacterial DNA is a circular genome, it won't knit back together, you need to actually patch it with some other DNA. And I was really interested in this idea of antibiotic resistance and could we repair it? So I discovered I sequenced the whole genome of a Staphylococcus aureus bacteria that lives on me. It lives in the front of my nose. And I've made a few artworks with this bacterium, and I learned how to whole genome sequence it. I learned how to do the bioinformatics, extract the DNA, grow it, everything, the whole, what they call pipeline of that research. And it has a penicillin resistance gene on its genome. And I was like, could I get rid of this resistance gene? So for years, this was nagging at me, how I could repair a genome. And the start of the antibiotic age is really 1941, which is when Florian Chain first trialled penicillin in humans. Now, we think of it as Alexander Fleming, who in 1928, spotted it in his lab, but he actually abandoned his research in 1929, saying it was too difficult. But he was quite the showman, and he liked to do the TV appearances and stuff, so people know him for the discovery of penicillin, but they don't necessarily know about Florian Chain, or even Dorothy Crowfoot Hodgkin, who also worked on the model of penicillin with them there, and is one of the few British women to have won a Nobel Prize for science. So it was a very important research. In 1941, they did the first trials of penicillin in humans in Oxford at the John Radcliffe Hospital, and they managed to keep the patient alive for about 3 weeks before they couldn't make enough penicillin. And they were biohackers because the war was on. It's an amazing collection it's another medical museum, the Museum of History of Science, or History of Science Museum in Oxford, and they have Florian Chain's fermentation vessels, which are biscuit tins with tin cans soldered on, bits of cotton wool stuff down them, which is what they used to grow this stuff because they couldn't get the lab equipment because the war was on. And so they were doing all this amazing stuff, and that was in 1941, and that's the year that the UK government brought out its Controlled Commodity 1941 utility mark, which was stamped on clothes, furniture, and anything that was deemed to be using the materials in a sort of sustainable way, if you like. But a kind of wartime sustainable way. So from wearing flary long skirts, it went to slightly tighter skirts and things like that in the clothing. And they brought out this leaflet shortly after, which was about repairing things, make do, so live with what you can live with, and mend, patch and repair. And it tells you things, unpick and knit again, hints on washing, decorative patches. So I was quite interested in this idea of the patching metaphor and the CRISPR DNA editing. So, and part of it came from, and this is the thing I was mentioning I made with Nicola Fawcett, I actually had to have a typhoid vaccination before I could make this work as well. Because these patches here, these decorative patches, it makes it very explicit the problem of antibiotic resistance in the gut microbiome. They are silk, which was grown with dilute fecal samples from patients in Oxford on petri dishes on what's called a brilliant UTI agar jelly, which turns, I think, Enterococcus grows blue and E. coli grows pink. And so different species grow different colours on it. So a healthy gut microbiome would be a nice combination of big and small pink and blue colonies. So this one's not too bad here, or this one's not too bad here. But you can see this person has only grown E. coli, and this one's Enterococcus, and this one's only grown E. coli. And so this is just different patients who have been on antibiotics and the damage it's done to their gut microbiome. So could we repair that using a technique of kind of patching? And so that came to this piece, which is an actual CC41 controlled commodity 1941 woman's suit that I have patched with silk that was grown with an E. coli bacteria that I CRISPR edited to remove an ampicillin resistance gene, an antibiotic resistance gene, and then I patched with the phrase make, do and mend, which was converted from ASCII code base 4 and written into the DNA and then chopped out the antibiotic resistance gene and repaired it with this phrase. So this phrase is now written into the genome of that bacteria, which was grown onto the silk patches, which are patching the dress. And in this bottle, you can't show genetically modified organisms very easily in museums and things like that because you need to have a license for the space. It can be done, but it's more complicated. But you can show the plasmids that you use to do it. So rather than showing the actual GM bacteria in life, I'm showing here, you've got the molecular scissors, the CRISPR molecular scissors, and on another little patch there, you've got the plasmid with the make, do and mend repair fragment on it. So if you combine that with the right strain of E. coli, then you can create your own CRISPR edit. And I did this all in the lab at Technion and worked with the scientists there. And the way that they do this is the protocol says you apply these things and you pick 20 colonies and then you test if it's worked. And there were other PhD students in the lab and things like that doing these things. And my experiment was the only one that worked. And the reason was, I think, because as I was from arts funding, there was no chance to repeat the experiment. If it didn't work, I would be making an artwork about how I tried to edit a bacterium, and it didn't work. I would be doing an artwork about that, but it did work. And the reason it worked is because instead of picking 20 colonies, I picked all the colonies on the Petri dish. So you have to get like separate colonies growing on your Petri dish. And each one, and then you test each one to see if it's taken up the edit. And one colony out of 60 had taken up the edit. You can see if I'd just done the normal protocol, it wouldn't have worked. So that's the difference between art and science. I think sometimes we just have to, like, sometimes maybe the lack of funding is better. In this jar here, this is antibiotic susceptibility disc. So each of these discs contains one antibiotic, one type of antibiotic. And so each one's one type of antibiotic, and altogether they are the entire antibiotic arsenal of the British Hospital. They're from the John Radcliffe Hospital. So that's all the choices of antibiotics we've got to go with. That's what we're dealing with at the moment. And this is it's a bit like, are we playing with this or what are we doing? So a toy sewing machine, but it's actually my mother's toy sewing machine that she played with in the war. But I've repurposed it and it's now stitching some of these modified bacteria together in this sort of allusion to patching and repairing.
So this looks absolutely fascinating. Tell me more about this.
So this is called disembodied dysbiosis, and it's based on a gut model which Jane Freeman, my collaborator on this, works with at the University of Leeds, so in the city. And they've got a room full of these things, and they start them off. So inside the jars that they have, the jars will contain fecal samples from a healthy donor. And they start them off and they grow the gut model, and it grows very happily, and it has these sort of biofilms that form on the inside of the jars, and the outside of the jar is like a water bath to keep it warm, and it has all these sort of different sensors and tubes and things like that going into it, and different gases into it and food going into it. So this is based on that. It uses an actual shelving unit from an old gut model from back in the day. Nowadays they're made out of metal usually, or plastic. Each of these jars is under different conditions, and it shows a different kind of part of the gut. So these what they do is they start it off and they have it growing all healthily. Then they will give it Clostridium or Clostridioides difficile, and that will start to colonise the jars. They're still perfectly happy because they haven't had any antibiotics, and it's just part of the normal human microbiome at that stage. And then they give it antibiotics, and then they get something called dysbiosis happening, which is where the biofilms that happily colonise the jars and the bacteria living in there, you get patches that come off, and it looks kind of raggy. So there's actually, it's kind of hard to see in the lighting, but there's actually, I've etched the glass on the inner jars, so you've got these kind of effects there. And then they study what happens, basically. It's how this works with this, what they call weir system, feeding each jar, and they're under gases, and it's bubbling away. But instead of these sort of lab sensors and things, I've made a representation of the sort of the lining of the gut made big, working with Alex May on it's 3D printed, so we 3D designed it. Sort of the kind of the folding of the gut, and these things which are like villi in the gut, and then they've got the cocci and bacillus bacteria colonising over it, and then it's all hand painted. So that's this, and I quite like this kind of the way it's almost sickly sweet. It's got that kind of, it's somewhere between a kind of, it's again, we were talking about this tension, and it's got this tension between a kind of beautiful, kind of intriguing thing and slightly disgusting thing. That's something that, you know, is something quite important to me. It almost reminds me in a way of ice cream. And a sort of with hundreds and thousands. So I was kind of thinking about that, also thinking a lot about bees, because it relates to this piece up here, which is called Fragile Microbiome, which is related to the name of the show. And these are, again, these patches of bacteria with holes in there, there's lots of holes there, which represents the dysbiosis. And these sort of circular things represent the villi, and you've got the bacteria clustering around them and in them. And these bits of linen lace, which have been grown in the gut model, are intertwined in some of them. I didn't make all the felted pieces. Some are made by the research group at the University of Leeds, with Jane Freeman's colleagues, and some are made by friends and colleagues. I did hand bead everything myself, so it was a long and painstaking process, and it is also impregnated with the extracted DNA of the gut model at Leeds University.
I love this piece, Anna. Tell me more about this.
This is called Viral Interventions, and it's a collaboration with Professor Maggie Smith at the University of York, quite nearby here. And she works with Streptomyces bacteria particularly, and also with bacteriophages. And phages are viruses that bacteria can catch. So people are always confused about what's bigger, a bacterium or a virus. Well, when you know that bacteria can catch viruses, it sort of puts it in perspective quite well, I think. And some people think that these phages could be a useful treatment to combat bacteria. So some people are researching that. It's kind of difficult to do because they're viruses, so they can evolve and mutate and change. So it makes it quite difficult to have it as a standard drug. But what Maggie's team were looking at when I was doing the collaboration was would these, is there an active ingredient, some particular molecule that's doing that stuff, and can we make that into a drug? So she was looking at that, and she was also looking at Streptomyces, which is a species of soil bacteria, or a group of soil bacteria, different ones. And this is Streptomyces silicolor, which produces a red antibiotic when it's feeling happy. And then when it's a bit like starving or hungry, it will produce a blue antibiotic when it's stressed. But she came up with a way, working with something called phage integrases, which is a precursor in a way to, or a slightly different method, but related to CRISPR, that it allows you to edit the antibiotic pathways of bacteria. So we can see this red here, which is the wild type in its happy kind of way, and they've edited the antibiotic pathway so it can only produce the blue bacterium. So this strain that was grown on here, so the bacteria is actually the blue dye, it's the antibiotic that's expressing in the petri dish has dyed the silk, as with the red here. And this cross here and this circle here that I've highlighted with metallic thread, that's actually painted with the bacteriophage, so with the virus.